what I'm seeing is in 25 years from now, a patient comes in with a disease and we are very quickly able to describe the disease or do the diagnosis at a genetic level or at a genomic level. And once we've done that, that we've got an ability to immediately pair up drugs to that particular subtype, such that the patient could get it.
I'm Jane Grogan, and I'm a scientist. I've been at this for more than 20 years now, and I think perhaps the only thing better than doing science is talking about the science. Lucky for me, I work in a place where I am surrounded by some of the brightest minds in research. However, there's usually not much time to just sit and talk. And that's why I'm so thrilled to be hosting this podcast. I am going to step away from my lab today and chat with a colleague about some of the cool stuff we're working on, especially as we try to link these discoveries to new medicines. So grab your favourite drink, get ready to unlock your science brain, and join us for Two Scientists Walk Into a Bar, a podcast for biotech geeks and the people who want to hang out with them. We all know medicines come with specific directions like how much to take and when. But have you ever thought about how research and researchers determine the dose?
Frankly, I have not thought about that.
I just rely on the information that's on the label.
I would assume, though, they would do it through the trials, kind of through trial and error to see the optimum amount that doesn't hurt people.
I don't know. I have no idea.
Now I'm thinking about it, right?
Today's focus is pharmacology, the branch of biology concerned with the study of drug action. This is a subject near and dear to my heart as I grew up as a pharmacist's daughter and then actually ended up majoring in pharmacology along with biochemistry in my undergraduate degree at the University of Melbourne. So what is pharmacology and how do we think about this in terms of possibly even personalized medicine and beyond? So with me today to discuss everything pharmacology and the science of pharmacology is Sara Kenkare-Mitra. Welcome, Sara.
Thank you.
Sara, let's start very simply. How would you define the science of pharmacology?
So pharmacology is a branch of biology, actually, and it's related to the study of drug action. It's everything about what the drug does to the body when you take a drug, and then what the body does to the drug because it has to take care of the drug.
And of course, this covers everything that's natural, found in the environment, constructed in a lab.
Yes. Everything. It could be pharmaceuticals, it could be natural products, it could be endogenous products. Any of these that has an effect and has to be handled by the body is pharmacology.
So in previous episodes, Sara, we've talked about chemicals and proteins and how they move around the body. And I'd like to kind of talk about how pharmacology fits into this, because I think a lot of people out there think a drug has a specific action, right? There's a problem that needs fixing and you provide a drug and it fits into that like a jigsaw puzzle and voila, it's all done. But in fact, the drug has to move around the body, it has to get to the site of action. It's not just as binary as I think a lot of people think.
Yeah, I think, with a drug, firstly, drugs are all different kinds. So let's say if we kind of broadly classify them as small molecules. So this is typically the drugs we take on a day-to-day basis. All of these are oral molecules. They're actually small molecules because they have a very small molecular weight.
And of course, most of these we take orally, ingest them as tablets.
And we take them orally, and they're either tablets or capsules. In case of kids, they may be syrups. Sometimes there are suppositories, etc. But these are drugs that are essentially taken by a route other than administering them directly into the bloodstream. Now, the other end of the spectrum are large molecules, and large molecules, which are proteins or antibodies, these molecules are typically given directly into the bloodstream. And with a lot of these drugs, you think it's all about just dosing it, and then it causes a certain effect, because that's what happens to a patient. You got a headache, you take an ibuprofen, and your headache goes away in a certain amount of time. But in between these two processes, a lot of things happens. Firstly, the drug that you take is, let's say, in a tablet form or a capsule form. It needs to first even get dissolved and absorbed and get into the bloodstream. When it gets into the bloodstream, typically the body sees it as a foreign molecule. It's or a xenobiotic, as we call it. And our bodies are built to destroy xenobiotics. So our mechanisms, such as our liver, and our kidneys will process these molecules. So you eliminate them from the body.
So really try to clear them out as quickly
Clear
as
them
possible.
out as quickly as possible. The liver is full of enzymes that are, ready to break these molecules up into inactive forms and get them out of the body. So under these circumstances, our fight is to make sure that the molecule is around for long enough time such that it can get to its real site of action. So the balance is between keeping that ibuprofen around in your body long enough so that it can actually do what it's supposed to do.
Jane,
That's Wellington, my producer.
can you clarify site of action versus when you talk about targets?
Ideally, you hope your site of action and your drug target are completely the same thing. That is, you have a drug that's targeting a molecule, that molecule is overexpressed in an inflamed or diseased tissue. That would be your site of action, so your drug would go there, hit the target at the site where it needs to work. Often things aren't as clean as that. Sometimes your target may be expressed on other tissue, normal healthy tissue, whether it be in the gut or elsewhere, and you hope that your drug either won't bind there very well, or if it does, it's not going to have a dramatic effect as it will when it's a concentrated amount in a tissue. So it's really understanding the dosing, how you can dose a molecule or a drug and keep it at a level that we know is required to drive efficacy.
Yes, so there's another factor in there to consider. We define drugs as drugs that have a narrow or a broad therapeutic window. A therapeutic window is the window at which the drug is safe, but as well as effective. And in some cases, that window is very broad. So you do not get side effects until you dose this drug at very high doses and very high concentrations. In the other situation, you've got drugs where the space between the dose at which the drug is active and the drug is safe is actually very, very narrow. Now, in these two situations, when you've got a drug that has these big swings, right? If it's in a broad therapeutic window situation, it's fine as long as it's not causing any harm and it's going to be effective. It could swing and still create the action it needs to create. But in a narrow therapeutic window situation, this is problematic.
So in the first example, typically then more drug doesn't necessarily bring along more side effects. You've got a large therapeutic window in which to operate.
That's right. Because even if you took a little higher dose, you still are very much in the safe range. And that's why the doctors, when they prescribe medicines, they will tell you what is reasonable and what is not reasonable. For example, please do not take more than 4 pills of this drug in a 24-hour window. The reason for that is if you do that, and if this is a narrow therapeutic window drug, then you could start seeing side effects.
So underlying the whole discipline of pharmacology is this concept of ADME, which stands for absorption, distribution, metabolism, and excretion. Could you just talk about why those different parameters are critical to the science of pharmacology?
And every drug is different. Some drugs are more fat soluble, so they go around into the fat, they stick around for a long time. Some just stick to your blood cells and remain in circulation, or the proteins in your blood and remain in circulation, and they never really go into your deep tissues. But it's still the same principle. The drug has to be absorbed, it gets distributed, and finally eliminated. So the ADME, understanding each of these and understanding that for a particular drug really helps you to design better drugs. It helps you to understand how your drug is going to work, where it's going to be, how long it's going to be there.
I think the excretion part is very interesting because you don't want the drug to be metabolized so quickly that it can't be efficacious, it's not there long enough. Yes. But at the same time, you may not want the drug to hang around too long. Like clearing it out of the system can be a very good thing. You want an effect, and then you want that drug to be cleared.
Absolutely. And I know many of the intravenously administered drugs actually They may have very short half-lives, but it doesn't matter. You can give them as an IV infusion, and you can get them to the concentration you need, let's say an antibiotic. And then once you stop the infusion, the drug quickly is gone from the body.
So, Sarah, when we think about the therapeutic window, it feels very discrete. And of course, everyone's different. Everyone's different in terms of their metabolism, whether it be the food that they're just eating at their meals. People are different sizes, different shapes, have different amounts of fat or non-fat, which is important depending on the kind of drug that you provide. How do you layer all of this complexity into deciding on a dose?
Yes, that's a great question. That is the key part of pharmacology, which is that every person is different. And so when I talk about any of these parameters, whether it's absorption, distribution, metabolism, elimination, half-life, clearance, all of this stuff, these are just average terms. But a really important job of a pharmacologist or a pharmacokineticist to now understand the variability associated with each of this. And with each of these steps, there's variability. There's variability in how we absorb drugs. For example, if you're taking an oral drug, if you ate it with one type of food versus another type of food, your transporters, there are transporters in our gut that are very different in different individuals. So the amount that gets absorbed is different. The amount that gets distributed is different. As you've said, some people have more body fat. Men are different from women. Different populations vary. Same is true for metabolism. In fact, there's an entire science related to drug metabolizing enzymes. And it is known that a lot of them can be polymorphic even, which means that there are multiple forms of even one enzyme. And in some populations, one form is more predominant, in another form is predominant. And that the drug metabolism then in that particular population might be very, very slow. They might be slow metabolizers. Another population might be a fast metabolizer. And really understanding that is very important.
Of course, the clinical trial is the empirical testing then, and then getting the endpoints that you can take back and understand that variability. But how do you model this up front?
So we typically try to start modeling this very early. Even when we are going into a phase 1 study, based on our preclinical information, we begin to develop models. These are computer-based mathematical models. And as we get more information from each stage of development, we add to these models. And those models are the ones that really help us better predict the pharmacology of the drug in different populations.
Jane, these models seem very complicated. How do they work? Are you just inputting data points for patient X and data for drug Y and putting them together?
This is largely a bioinformatic approach. It's taking information, putting it into a machine, and using machine learning to try and come up with some outputs. It's very important. It's early days. We'll see more of this in drug development moving forward. And this, of course, complements other research that goes on that's a lot more empirical or hypothesis-driven. How do you and the field think about identifying the right pathways for a given individual so that you and I, with a similar disease, could be taking very different doses and or different drugs?
There's pharmacogenetics and pharmacogenomics. And what it is that we clearly understand that there's a lot of variability. Now, not all variability is genetic in nature or has a genomic component to it. Some of it is just based on things like age or body fat percent or weight or things like that. But once you've ruled out all of those, then if there's a significant variability still remaining that you notice that a certain population has it, then looking for potential genetic drivers for it is where the field is obviously going.
Could you just take a moment and describe the similarities and differences between pharmacogenetics and pharmacogenomics?
Yeah, these two words are usually used interchangeably. I think when it comes to ADME, they're still a bit interchangeable because we don't have enough examples of pharmacogenomics. Pharmacogenetics is where you've got one particular gene that you know that potentially could have a direct connection with some effect or change. Pharmacogenomics is where you look at the entire genome or a portion of the genome and then try to relate it back to either efficacy or safety or something like
So
that.
if there's any, small genetic mutations that are actually affecting gene transcription or a role it may be having in metabolism or...
Yes, that those would show up in the pharmacogenomic screen versus pharmacogenetics is where you've got a hypothesis and you go in with a particular genetic hypothesis. And there are good examples of that already in the clinic or in practice. I was thinking about the drug. There's a HIV drug, and this drug used to produce a lot of hypersensitivity reactions in patients. And this went on for a while until they figured out that there was a particular type of HLA gene that once identified, if you found that patients have that gene, and you can reduce or either not give that drug, and it completely changed how the drug is dosed because you reduce the hypersensitivity reactions
eliminate that
and
toxicity.
eliminate that toxicity. So that's the future that we are hoping for and that we are all striving for.
Yeah, so do you think understanding the pharmacogenetics and pharmacogenomics aspect of pharmacology will add to our need for biomarkers or our use of biomarkers? We have talked about biomarkers in this show previously. Do you think this whole area of pharmacogenetics will feed into how we think about diagnosis or placement of drugs?
Yeah, I think that this field actually is more evolved than the field of pharmacogenomics as it relates to ADME or pharmacokinetics. And that's because particularly, in certain areas like cancer, the disease and how the disease is evolving and how heterogeneous it is and how well it's characterized, that has gone quite far. So we already have a good basis of genomics. Whether that will now play into pharmacogenomics and our understanding of pharmacology is still yet, the jury's out on that. But clearly we have a lot more information about cancers, about their heterogeneity, about their genomic information. And a lot of the work that's going on around the world is really to be able to relate that genomic information and connect it to figuring out which drug will work best for that particular disease.
And how far are we away from this? Is this a 5-year goal, a 10-year goal?
We already have a few
aspirational goal.
Yes, we already have a few examples where this is working. So we are not. This is not something that's so futuristic. We have a few drugs on the market today where you can pair up the particular genomic information or the type of cancer to a particular type of drug. So that exists. We would hope that there would be more progress in this area. A part of it might just be our own understanding of the biology is evolving, the technology is evolving, and I really think that in the future, this is going to be the state of the art. It may be 10 years from now, it might be sooner, but it has to be. That's where this field is evolving.
I want to ask you, what drew you into pharmacology as a student yourself?
When I was young, this starts very young because my grandfather was a physician. And so the talk in the house, and he lived with us for the early part of my life, and the talk in the house was always about ailments and medicine. I just cannot even remember a day without that. In fact, I almost would pretend doctor most of the time in the house if anybody got sick. And I just always was fascinated by medicine. Medicine was it. I could not speak beyond medicine. When I went into college, it was a toss-up between wanting to go to medical school or entering pharmacy school, where I could actually study more about medicine. So I was in pharmacy school. As part of my pharmacy school education, I had to do an internship. And I was an intern in India. And I was just thrilled. It was like my first true job. I was given a job to be in the tableting department. And my job was to sit in, the whole room was full of machines that were doing tableting. My job was to sit there and then occasionally get up and do certain tests on the tablets that were coming out. These were part of their quality control. And I was doing this day after day after day. And if you've ever been in a room with tableting machines, there's a constant noise like tarak, 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 tarak. And this went on for a month. And there was like this moment where I thought, what am I doing? While this is what, I love, that's not the aspect of it that I love. It's not the aspect of making that drug eventually. It's the aspect of understanding the science that leads you to what the drug is doing, and then making the drug work. That's the piece that excites me, not this. So this was my true aha moment when I decided that I have to get into graduate school and I have to pursue science beyond this.
So knowing what goes into the pill rather than just making the pill. For the younger people out there listening to this podcast and who are interested in this discipline, what advice would you give to them?
Well, firstly, my first advice is I'm just happy to let people know that such a discipline exists, because sometimes people even coming into science may not realize that there's actually a discipline around pharmacology, and it's a distinct discipline that's linked to biology, and that if you're interested in treating disease and you're interested in drugs and creating new drugs and drug action, then this is the place for you.
Since you began as a student, what have been the major advances in the field that have kept you really excited about the discipline?
If you looked at history books and how pharmacology started, it was all around, dosing, giving someone a certain chemical and then seeing if they actually, produced a certain effect or not. Over the years, science has evolved so much that our ability to actually measure things, like measure the drug, measure where it's going in the body, our understanding of drug metabolizing enzymes, our understanding of transporters, our ability to determine, to model all of this. A lot of the technology that has allowed us to then gain a more deeper understanding, go from empirical to really a scientific understanding of how pharmacology happens, is where we are today. The other area it's evolving is the mathematical modeling portion. And the reason I say that is because it's not always possible for you to test every scenario in the human being. But models that allow us to use this scientific information in a computational sort of way to then predict what the behavior of a drug would be in certain specific situations are already, paying a lot of dividend in this field. And in the future, this is going to be incredibly important. We are already seeing that there are people that are developing apps around this that might allow physicians to predict, how a particular drug would work in that particular patient that's in their office at that time. So one could imagine that this becomes incredibly more tailored than it is to this day.
And aside from the empirical and the computational, how much can be done in silico? How can we model any of this in a plastic dish? Can we grow a liver and look at metabolism in a dish without having to go into the patient early on?
Yes. So
kind of crazy ideas, but
Yes. So, there is a lot of work already ongoing in terms of using cells, for example, that will allow us to predict these type of behaviors, drug metabolism, etc. Using artificial organs, I think that's coming, right? Our ability to then being able to understand that is very important. However, one of the big, for me, the ahas in pharmacology is sometimes these isolated experiments may tell you a story that's not yet complete until you put the whole picture together. But yes, being able to, drill down and use, for example, artificial organs or in silico methods is incredibly important. And this is why the field has made the progress that it has.
I think that's a challenge in all biology, actually. It's the reductionist. How do you reduce the system down to understand it and then build it back into the complexity in which it exists?
Absolutely. Yeah.
Reverse translation question, Jane. In what ways will this work impact basic research?
Loads of ways. Research is an ongoing inquiry into uncharted or unknown areas. And we don't have all the answers. If we did, we'd have the cures, we wouldn't need any more drugs, etc. But we don't. And I think as we understand the complexity of human disease, the toxicities associated with drugs, we understand what works and what doesn't, we can actually take that and create hypotheses back in the lab and start asking why and how, and maybe coming up with a better drug or a lower toxicity profile of an existing drug that we can then take back to the clinic and to the patient. Sarah, Star Trek question. 25 years from now, a patient comes in with a disease. How could this look that's very different from today in terms of speed of diagnosis, speed of getting the right drug at the right dose with the right, therapeutic window profile?
Yeah, that's a fascinating question. So what I'm seeing is in 25 years from now, a patient comes in with a disease. Because of our understanding of genomics and genetics, we are very quickly able to describe the disease or do the diagnosis at a genetic level or a genomic level, and be able to make sure that we understand the exact subsets of this disease in a very, very rapid manner. And once we've done that, that we've got an ability, either through apps or more digital sort of methods, to immediately pair up drugs to that particular subtype, such that the patient could get it. I also think that there will come a time when the patients also have some profiles which allow us to understand, their cytochrome P450 profile, their transporter profiles, their elimination profiles, etc. And all of that is available already as part of a health chart.
So maybe it's more than just coming in and knowing the blood type. It's knowing how you will metabolize something.
Absolutely. And that's already in your medical health record, and it essentially you can use digital information to then pair this, because you've got the entire, the entirety of the patient as an individual, as well as their disease, the patient and their disease, and those are very well classified. And now you've got drugs that you can pair based on that combination, rather than the more, trial and error methods that happen at this point in time, because they're based on the population rather than the patient.
Sarah, you've been awesome. It's been great. I think it's a really exciting time for pharmacology, especially when we start combining it with genetic information and all the computational and digital power that's out there these days as well. So thank you for sharing your insights.
Great. Thank you for having me here, Jane.
Lots to think about when you next reach into your medicine cabinet. As we get closer and closer to a world of personalised medicine, I think we will be increasingly reliant on the work of Sarah and her colleagues in pharmacology to ensure we are safely taking the right amount of medicine at the right time for our individual needs. In our next episode, we will be taking an even deeper dive into the immune system and exploring how the same system that protects us can also be the source of harm. You won't want to miss it. In the meantime, keep telling your science fans about us, like us on Facebook and Twitter, and most importantly, if you haven't already, subscribe and rank us on iTunes. Those of you who have already subscribed, thank you. And now for me, it's back to the lab.